Okay, thanks very much. I think they scheduled me here because I'm the transition shock between the chemistry and the biology that comes after this. And so today I'm going to kind of switch gears to think about microbiology and some of the work we've been doing in my lab in the University of Guelph with John Gallero, who has been working for me. And it started off with Tandra Fraser, who did her PhD with me and now is at the University of Reading. And we're working on some of the Leochondrin soils in New Zealand. So we've been thinking about non-specific phosphatases, and these are enzymes that show activity towards several different types of unrelated phosphoesters, so targeting phytin, nucleic acids, and phospholipids. And this makes them a really broad category of enzymes with, and challenging to target with molecular methods just because they're so diverse. And we all know that there's acid phosphatases and alkaline phosphatases. And where we live in Guelph, our soils are mostly calcareous glacial tills, and so these are all alkaline soils and well buffered. And so we started our work looking at alkaline phosphatases in soils. And Tandra started her PhD and looked at a field site where we had long-term organic a long-term organic trial at Glenlee, Manitoba, and the pH of the soil was 7.4, and she was measuring phosphatase enzyme activity, and we wanted to target the genes at the same time. And there was some primers published already by Tan et al. In 2013, and we just used those primer sets for PHOD, and they worked quite well in our soils. So we were able to link acid phosphatase activity with bacterial PHOD quantities and abundance in the long-term field trial. And then in a greenhouse study, we also linked gene expression and abundance of PHOD with the alkaline phosphatase activity. But Tandra actually did probably 5 times as much work that she needed to do for her PhD. So once she defended her thesis, she still had actually another 2-year field trial that didn't actually make it into her thesis. So once she left my lab, we thought we should write this stuff up. And she had this 2-year field trial where she looked at organic soybean farms across Prince Edward Island. And so we were interested in looking at, again, the gene expression and gene abundance in the systems, but the pH was lower, so between 5.1 and 6.3. And so we were concerned that the PHOD gene wouldn't be appropriate in this case. And so at that time, I hired a student, John Gallero, and he came to my lab and was interested in working on this system. And so what we decided to do was try to come up with some acid phosphatase gene primers. And we really were interested in primers that we could use for quantitative PCR so we could quantify gene abundance and also look at diversity. So that's where he started. And lucky for me, within a week or 2 of him starting in my lab, he actually had to have shoulder surgery. So he was stuck and couldn't do any lab work. So I put him in front of the computer for a few months, and he just dug through NCBI databases for gene sequences. And he came up with an amino acid-based phylogeny. And this was actually based on a paper by Martinez in 2007, who he started from there. And so we looked at amino acid sequences, and these are some of the ones that we started with. And these acid phosphatase genes are grouped according to their conserved structural motifs. And so talking to Tandra and John, we were thinking maybe we could come up with primers for each of those structural groups instead of trying to come up with one primer set for all of the acid phosphatase genes. And so this is actually the hardest part of this talk for me, because I was trying to figure out each of these classes and how they're defined in the literature. And if anything comes out of this workshop, if we could come up with some kind of common nomenclature for the genes involved in each of these classes, that would be great. So the first class is the class A, and they are secreted proteins. They're sometimes called nonspecific acid phosphatase group 1. In the literature, they're PHO-C and PHO-N genes. And the second group are the class B, and they're metalloproteins. Some are periplasmic. The genes are APPA and APHA, and they're also sometimes called the E. coli nonspecific active phosphatases because E. coli is a major group in the class B. And the third class are class C, and they're lipoproteins, and I've seen them to be described as secreted and membrane bound. And they're sometimes called non-enterobacterial acid phosphatase group, and I've seen the genes OLPA described. There's also plant acid phosphatases in archaeal, but we wanted to target the bacterial ones to start with, so we just developed primers for class A, B, and C. And so when we took these primers and tried them in the Prince Edward Island soils, what we found was we could quantify PHOC or class A phosphatases in these soils. So we had 12 field sites, and we found that they were much higher than the PHOD genes in this system. So that was good. And we could only quantify class A in these soils. We weren't able to quantify class B or class C. So we did look for a correlation between phosphatase activity and the gene copies, and interestingly, we saw that in the bulk soils, there was a correlation between PHOC and phosphatase activity. But in the rhizosphere soils, we didn't see that correlation, but we saw a correlation between the PHOD genes, which is the alkaline phosphatase, and the phosphatase activity. So that was kind of interesting. But at this point, we were really kind of curious about our primer sets. In silico, they should have worked and picked up these genes in the soil, so we didn't know if it was the soil or why we weren't picking these genes up in these soils. So we reached out to Leo, and we looked at one of his field sites in New Zealand that has a pH of 5.6 to 5.8. And Gustavo's going to talk about this a bit more, but we started with his long-term ecological trial, and this was established in 1994, so it's been going for 22 years. And so there's a bunch of treatments within this trial. There's a mowing treatment and a fertilizer treatment, and then there is clippings or residue returned and removed treatment. And so the ones we targeted were just with mowing, 0 nitrogen fertilizer, and then the comparison was residue returned or removed. So I grabbed this off Facebook, which Leo's not too happy about, but he went out to the field and sampled soil. And I think Gustavo might be in the back there, in the background. This is the other trial we sampled. But, and then once he sampled that soil, he put it into tubes for us that contained a lifeguard solution, which stabilizes nucleic acids in the field. And then he couriered it to Guelph, which was probably the most difficult part of this whole experience. And we got it in my lab a little while later after some issues with customs, and then we extracted DNA and RNA in my lab in Guelph. So when we looked at bacterial populations just with 16S, you saw a slightly higher population in the plots that had residue removed, but there wasn't any really significant difference. And in some papers earlier on from this site, you see that there's a community structural difference between these two plots, but just quantities of bacterial populations, we didn't see any differences. And then when you start looking at the acid and alkaline phosphatase genes, so the A, B, and C are the 3 groups, and then the PHOD is the alkaline phosphatase. So you'll notice even I changed nomenclature during one talk. Anyway, so what we looked at is the gray bars are abundance. And we did find all 3 genes present for the 3 alkaline phosphatase classes in these soils, and also the PHOD. And we saw generally they were higher in the plots that had the residue removed, and for the class B, they were actually significantly higher in the residue removed plots. And the lighter gray bars is gene expression, so looking at RNA, and we were able to quantify gene expression in the class A, B, and PHOD. And in all cases there, gene expression was higher in the residue removed plots. And so When you look at OLSEN P in these systems, the biomass retained plot has significantly higher OLSEN P than the biomass removed, about almost twice as much, I guess. And so this kind of made sense for us. So where there's lower phosphorus available, you start to see gene expression and higher phosphatase gene activity. It gets a little more complicated when we look at the acid phosphatase numbers in this system because the residue return plots have higher acid phosphatase values than the residue removed. So that's been a slight complication. But basically what we wanted to do is link acid phosphatase activity, OLSEN P, and these nonspecific acid phosphatase gene copies. And so if you look at the system, in the biomass retained plots where the clippings are returned, you have the higher OLSEN P and slightly higher acid phosphatase. And on the biomass removed, you have lower OLSEN P and slightly lower acid phosphatase. And so when we looked for a correlation at the beginning, we saw no correlation between the gene copies and the acid phosphatase values. But when you separate the data into the plots that have the residue removed and the residue retained, you start to see correlations between these two groups. So this is just the Class B data, but you can see there's close to significant correlations in the two when you look at them individually. And so if you looked at all three classes individually, they all correlated about the 10% level. So this is suggesting to me that these two systems probably have two different microbial communities associated with them that might be acting differently and expressing different acid phosphatase genes. And so the next step for us, and John's back in the lab right now, is doing some sequencing with high throughput sequencing using Illumina. And what we'll do is, hopefully with this data, be able to tell whether just how well our primers are covering these genes and how specific they are, and also whether the communities of these acid phosphatase containing genes are actually different between the two systems. So with that, this funding has been kind of piecemeal, but it's supported by Canada Research Chairs Program and NSERC, and also a little bit from the Ontario and Agriculture Canada. So thanks very much.